Hello everyone and welcome to the new session of the lecture series, and today's topic is cell cycle and cell division. And within this topic we'll be studying about the stages of mitosis in detail. So let me first give you a brief introduction of the topic. Now it is already known that the cells, they arise from the pre-existing cells, and the process of this formation of new cells, this process is known as cell division. This cell division leads to the growth and development of the multicellular organism. Alright, so the cell division leads to the development and growth of any organism. So let's talk about cell cycle now. Now, how can we define cell cycle? So cell cycle is simply the stages through which a cell passes during the cell division is known as cell cycle. Now, cell cycle is basically divided into two major phases. First one is the interphase, and second one we call it as mitotic phase, okay, also known as the M phase. Now, let's talk about the interphase. So interphase is further divided into three phases. First one is the G1 phase. Okay. G1 phase is also known as the gap 1 phase. And it is also known as post-mitotic gap. Why it is called post-mitotic gap? Because it is happening after mitosis here. That's why post-mitotic, after mitosis. Second phase of interphase is synthetic phase, also known as the S phase. And third one is the G2 phase, also known as the gap 2 phase, or we can also call it as pre-mitotic phase. Means it happens before mitosis. Here you can see G2 phase here in this diagram. This is the G2 phase. This is happening pre-mitosis, before mitosis. Okay, another phase of cell cycle is the M phase, also known as the mitotic phase. The mitotic phase is further divided into two phases. First one is the karyokinesis, in which what happens in karyokinesis, nuclear division takes place. Division of nucleus takes place. And second one is the cytokinesis. Cytokinesis means cytoplasmic division. Here you can see mitotic phase. This one is mitotic phase. From here till here it is mitotic phase. It is divided into karyokinesis and cytokinesis. Alright. Now let's talk about interphase in detail. So interphase is also known as the resting stage, and it is a period of synthesis where growth is taking place. There is a lot of synthesis taking place, and interphase we all know now is divided further into G1 phase, S phase and G2 phase. Here you can see this is a diagram showing the interphase, and it is divided into three sub-phases. First one is the G1 phase. Okay, then we have the S phase, and then we have the G2 phase. After G2 phase, we have the mitotic phase, okay, which is divided into karyokinesis and cytokinesis. And ultimately what happens, there is a formation of two daughter cells in the end, after the end of mitosis. So interphase is the longest phase of the cell cycle, and it comprises about 95% of the duration of the cell cycle. So the cell spends most of the time in the interphase stage of the cell cycle. Let's talk about the sub-phases of the interphase. First one is the G1 phase. So G1 is the phase between the mitosis and the DNA synthesis stage or the S phase. In S phase, DNA synthesis takes place. That's why we are calling it as DNA synthesis phase. So now we know that interphase was divided into G1. Okay, then we had S phase. We had G2 phase. Okay, and then we had M phase. So G1 phase is between the mitosis, okay, and the S phase. And what happens during the G1 phase? During this phase, the cell is metabolically active. We say that interphase is a resting phase, but the cell is actually metabolically active. It is preparing for the cell division. And what is happening in G1 phase? There is a lot of production of carbohydrates, lipids, RNA and proteins. Okay, so what happens to the DNA content? There is no change in the DNA content here. There is only production of the organic molecules such as carbohydrates and all, as well as there is synthesis of RNA as well. But there is no synthesis of DNA. So what happens? Sometimes what happens during the G1 phase, this is a point to remember, during G1 phase, what happens? Some of the cell, they stop dividing, okay, and they remain as such. And they get arrested in the G1 phase of the cell cycle, and then this stage is known as the G0 stage or the quiescent stage. So what happens if this is the G1 phase? We have the S phase, we have the G2 phase, and then we have the M phase. Sometime what happens, cell cycle is going like this, from G1 to S, then to G2, then we have M, mitotic phase, and then we have the G1 phase. So sometime what happens, some of the cells, they stop dividing and they remain or they get arrested in the G1 phase here. Okay. And this stage is known as the G0 stage or the quiescent stage. Now let's talk about the second subphase of interphase, which is known as the S phase. S phase is also known as the synthetic phase. In synthetic phase, what happens? This is the unique feature of synthetic phase. DNA replication takes place here, as well as histone synthesis takes place here. And the other thing which is happening during the S phase is there is a duplication of the centrioles. Okay, now what are centrioles? Centrioles are cell organelles that facilitate and they help in the organization of the spindle fiber. So here you can say they facilitate the formation of spindle fiber. Formation of the spindle fibers. And what are spindle fibers? Spindle fibers are nothing but long cylindrical proteins made up of microtubules. Then after S phase, we have the G2 phase. Okay, so G2 phase is a period between the S phase and the mitosis phase. Okay, we have again G1 phase, we have the S phase, we have the G2 phase and the M phase. So G2 phase is a period between the S phase and the M phase. What happens in the G2 phase? There is final preparation of the cell division takes place here. Okay, cell is preparing itself for the cell division. And what is happening? The cytoplasmic organelles such as Golgi apparatus, okay, mitochondria, they are doubled. Okay, another thing that is happening here is the synthesis of the tubulin pigment, microtubule formation, okay, which are required for the formation of the spindle apparatus. Or you can say formation of spindle fibers, because spindle fibers are what? They are made up of microtubules. Now about the quiescent stage, we have already talked about the quiescent stage. So what is happening? Sometimes what happens, the cells get arrested at a specific point in the G1 phase. So if this one is the G1 phase, the cells get arrested in the G1 phase here. Okay. And the cells, what they happen? Some of these cells, such as heart cells, or you can say cardiac cell, skeletal muscle cells, some lymphocyte and some nerve cells, what they do? Sometimes they get arrested in the G1 phase and they are unable to re-enter the cell cycle. So these cells are then said to be terminally differentiated, means they are arrested in the G0 phase. And what happens later? When conditions change, the growth is resumed and the cell re-enters the G1 phase. Okay, so depending on the conditions, okay, if the conditions are favorable, the growth will resume and the cell can re-enter the G1 phase and then it will follow the S phase, G2 phase and the mitotic phase. Now, the enzyme responsible for the arresting of the cell in G1 phase. Okay, this enzyme, the name of the enzyme is cyclin-dependent kinase. So this enzyme is involved in the arresting of cells in the G1 phase. Okay, no need to go in detail about the mechanism. You have to just remember that enzyme responsible for arresting the cell in G1 phase is cyclin-dependent kinase. And what is the significance of this G0 state or quiescent state? The significance is that it helps in the regulation of the duration of the cell cycle. Okay, so cell cycle is regulated with the help of this quiescent stage. Now let me introduce you the term C-value paradox. So how can we define C-value paradox? C-value paradox is nothing but it is the total DNA content of the haploid set of chromosomes. Or you can say C-value is the amount of DNA per haploid cell. Okay. So haploid cell, if the number of chromosome is N, okay, then the DNA content will be 1C. Because the DNA content is equal to the haploid sets of chromosome. Okay, that's why if the number of chromosome is N, DNA content will be 1C. If the number of chromosome is 2N, okay, 2 chromosome sets are present, then DNA value will be 2C here. Okay, so this is for diploid cell, it is 2C. This is for diploid, means 2 set of chromosome. This is for haploid cells. So the amount of the DNA is usually expressed in picograms. Alright, so DNA content here of the germ cell. So germ cells are what? Germ cells are haploid. They have 1 chromosomal set. They are haploid. So the DNA content will be 1C. Okay. And the DNA content of somatic cells, what are somatic cells? They are diploid. They are 2N. Somatic cells, also known as vegetative cells, these cells are diploid. Okay, so in somatic cells, the DNA concentration will be, or the DNA value will be 2C. But what happens? Initially, the DNA value is, or the DNA concentration is 2C. But what happens after the S phase? Now the cell enters the S phase or the synthetic phase. And what happens during the S phase? There is DNA synthesis or DNA replication takes place. Now what happens? The DNA content is doubled here. Now the DNA concentration will be 4C. Okay. But the number of chromosome or the chromosome sets are 2 only. So it will be 2N but the DNA concentration will be 4C. Here what I have written that DNA content in the somatic cells during the G1 phase will be 2C. But after the S phase, after the DNA replication, the DNA content become 4C. But the chromosome number or the cell will be in diploid state only. It will have 2 chromosomal sets. So we will represent it as 2N. Now let's talk about the nucleus. Let's talk about the condition of the nucleus after the interphase, or we can say at the end of the G2 phase. So what is seen or what is observed, that each chromosome exists as a pair of chromatid joined together by a centromere. Okay, now what is a centromere? Now, centromere is a constricted site present on the chromosome. It is a constricted site present on the chromosome. And this is the area or the region where the spindle fiber attach. Okay, so how is the chromosome visible at the end of the interphase? Chromosome consists of 2 pair of chromatid. So chromosome appear like this. Okay. In the center we have the centromere here. Here is a centromere where the spindle fiber will attach. These are the chromatids. Or we can say these are the sister chromatids. These two. Okay, so the chromosomes are visible as pair of chromatids joined at the centromere. Cell is in diploid condition here. Okay, because mitosis usually takes place in diploid cell. The cell is in diploid condition, but the DNA content is double because cell has passed through the S phase and the DNA content is 4C instead of 2C. Before S phase it will be 2C, after S phase it will be 4C. Another thing here to remember is that the chromosomal material is in the form of very loosely coiled chromatin. So chromatin network is visible. So if this is the nucleus, chromatin are visible like this. There are thread-like structures. So this is chromatin. Thread-like structures. And then we have suppose this is the nucleus, nucleolus. This is the nuclear membrane. This is the nuclear membrane. Alright. So chromosome is visible as chromatin like network, which are nothing but thread-like structures. The centriole is replicated into 2. Another thing here, what we can observe, this centriole has been replicated into 2, means 2 centrioles are present. Now what is a centriole? Centriole is nothing but it is a cell organelle that facilitate and help in the organization of the spindle fibers. Or you can also say centriole help in the assembly of the microtubules. Now let's talk about the M phase, okay, which can be mitotic phase or the meiotic phase. About meiosis we will talk about in the later stage. Today our focus is on the mitotic phase. So mitotic phase is the actual dividing phase, okay, and it is the shortest phase of the cell cycle. Whereas interphase was the longest phase of the cycle that comprised about 95% of the cell cycle. Now mitotic phase is further divided into two subphases. First one is karyokinesis. Second one is cytokinesis. Karyokinesis is nothing but division of nucleus. Cytokinesis is nothing but division of cytoplasm. Now mitosis is known as the equational division. So let me introduce you to the topic, to the term mitosis. So the term mitosis was coined by Fleming in 1882. And when we talk about mitosis, we usually talk about the diploid cells. Mitosis usually occurs in diploid cells of animals and plant cell. Okay, but there are few exceptions such as haploid male drones of honeybee. So the male drones of honeybee are haploid, means they have one chromosome set. Okay, and another example is, another exception is haploid cells of lower plant. In male drones of honeybee, what is happening? So they are haploid and mitosis takes place in the male drone during the formation of sperms. Sperms have haploid number, so sperms are formed because of mitosis in honeybees, in male drone of honeybees. Okay, so another point here is that mitosis leads to an increase in the number of cells without any change in the genetic makeup. Okay, so the genetic makeup of the offerings remain the same. The daughter cells which are produced, okay, they are qualitatively and quantitatively similar to the parent cell. Okay, so their genetic makeup remains the same. Now let's talk about the karyokinesis stage. So the karyokinesis stage is further divided into prophase, metaphase, anaphase and telophase. First, we will be talking about the prophase stage. So the prophase is the first stage of the karyokinesis. And in this stage, what happens? There is a condensation of chromosome takes place. Okay, so this is the diagram showing the prophase. In this, what you can see, condensed chromosomes are seen here. These are the condensed chromosome. The chromosomes have two sister chromatids. Okay, these are chromatids, sister chromatids. This region is the centromere, which consists of two kinetochores, centromere with attached kinetochore. Now, what is a kinetochore? So, these circles you can see, these are kinetochores. So, kinetochores are what? Kinetochores are protein complexes. Kinetochore, these are nothing but protein complexes present on the centromere. And this is the region where the spindle fiber will attach. Another thing happening in prophase is, this I've already told you that each chromosome have two chromatids held together by a centromere. And another thing happening in prophase is that the cell organelles like endoplasmic reticulum, Golgi bodies, nucleolus and the nuclear envelope, they start disappearing during the prophase. There is condensation of chromatin is visible. Okay, and each chromosome consists of two sister chromatids connected by a centromere. Now, centrosomes are present in the cells. These centrosomes, they begin to move towards the opposite poles. And what is happening? So, if this is a cell, centrosome, okay, they move to the opposite poles. And what happens? Then they radiate the microtubules. Microtubules are involved in the formation of spindle fibers. Okay, so they radiate microtubules. They radiate microtubules from them. Okay, and this structure is known as aster. The radiating microtubules are known as aster. Here also it is known as aster, A-S-T-E-R, aster, and two asters together with the spindle fibers later form the mitotic apparatus. Now the second stage is the metaphase stage. In metastage, what happens? Condensation of the chromosome is completed. And what happens? The chromosomes are arranged at the equatorial plate. This is the metaphase plate, also known as the equatorial, means equator. Equatorial plate, or you can say equatorial plane. Here you can see the chromosomes lie along the equatorial plane, also known as the metaphase plate. These are the spindle fibers. Okay. Here is the cytoplasm of the cell. These are the centrioles, and asters are seen radiating from the centriole. One point here to remember is that at metaphase, the morphology of the chromosome is most easily studied. Okay, so the chromosomes are visible very clearly at the metaphase stage. And at metaphase stage, what is the condition of the chromosome? The metaphase chromosome is made up of again two sister chromatids which are held together by the centromere. The chromosome, we know, lying at the equator at the metaphase plate. Okay, and the spindle fibers are attached to the kinetochore of the chromosome. And what was kinetochore? Kinetochores are protein complexes present on the centromere, and they are the region where the spindle fiber will attach. Now, next stage is the anaphase stage. In anaphase, what happens? Two things are happening here. First one is the centromere division takes place. Okay. So chromosome was, there were two sister chromatids and the centromere was present in the center. So the centromere divides. Okay, this was the centromere. This centromere will divide. Okay, and the chromosome will split and migrate towards the opposite pole. Okay, so this was the chromosome with two sister chromatids. So one chromatid will grow here to one pole, another chromatid will go to the other opposite pole. Okay, and there is this is because of the division of the centromere here, and that's why it is called as the equational division. Okay, so what is happening here? Chromosome break at the centromere and chromatid moves to the opposite poles here. This was one chromatid, this was the other chromatid. Both the sister chromatid, they move to the opposite poles. And what is happening? The two daughter chromatids that are now separated, now they are known as daughter chromosome. So instead of saying daughter chromatid, now we say them, or we refer them to as daughter chromosomes. Okay, so these ones are daughter chromosomes. Now the last phase is the telophase, which is also known as the reverse of the prophase. Why it's called reverse of the prophase? Because all the things happening during the telophase are just opposite of the prophase stage. So what is happening in prophase? Chromosomal condensation of chromosome was taking place during the prophase, but in telophase, chromosome, they decondense and they lose their identity. The nuclear envelope was disappearing during the prophase, but here nuclear envelope, it start reappearing around the chromosome. Okay, and later on, two daughter nuclei are formed. So here you can see there is a formation of nucleolus, there is formation of the nuclear membrane. The cell organelles also start appearing here. This one is the nucleolus. Nucleolus is what? It is a membraneless structure present inside the nucleus. Alright, so these are the two daughter cells. This is one daughter cell. Okay, this is the second daughter cell. There are two daughter cells are formed here. which have the same genetic makeup. That's why we call it as equational division. Now, we have already talked about this, that the nucleolus, Golgi bodies, or we can say Golgi complex, endoplasmic reticulum, they reappear during the telophase. Okay, so all the stages here which are happening are reverse of the prophase. Okay, so this was the end of the karyokinesis. Now, after karyokinesis, another stage is the cytokinesis, which lead to the formation of two daughter cells. In cytokinesis, what happens? There is a cytoplasmic division, which leads to the formation of two daughter cells. Two daughter cells will be formed. Okay, so there is a difference between the cytokinesis in animal cell and the plant cell. So let's talk about first the mechanism of cytokinesis in animal cell. Okay, so the mechanism is called as the cleavage furrow method. In cleavage furrow method, what happens? A contractile ring, initially, which is made up of actin and myosin filament, it appears at the equator of the cell, or the equatorial plate, or the equatorial plane of the cell. What happens? Then this contractile ring, it shrinks at the equator. Okay, it shrinks, it moves inwards, okay, pinching the plasma membrane inwards. And what it does, as it moves inward, okay, it forms a cleavage furrow. Here you can see, this was the cell at the telophase. So this was the cell at the telophase. This was the cell at telophase. Okay, this is the contractile ring which was formed at the equator. This is formed at the equator. This contractile ring, it moves inwards, okay, pinching the plasma membrane. It is moving inwards here, forming a furrow, and as it moves inward, it forms a furrow. And what happens later on? What is happening here? It shrinks to a point that there are two separate cells are formed, okay, which have their own plasma membrane. So what is happening? Eventually, the contractile ring shrinks to a point that there are formation of two separate cells in the end, and both the cells have their own plasma membrane. So briefly, what we can say, that there is a formation of a contractile ring which is made up of actin and myosin filaments at the equator. This contractile ring, what it does, it moves inwards, pinching the plasma membrane, and as it moves inwards, finally or eventually what happens? It leads to the formation of two daughter cells. Okay, these purple one are the chromatic network, then we have the vesicles here, and these are the microtubules. Okay, these are the microtubules. Now what is happening? What is the mechanism of cytokinesis in the plant cell? Okay, in plant cell, the cytokinesis is happening by cell plate formation. So what is happening in the equatorial region? Spindle fibers, initially they create a barrel-shaped region. Okay, so this is the barrel-shaped region which is formed at the equatorial region, and this is known as the cell plate or the phragmoplast. Alright, so what is happening here? In this region, in the equatorial region, what is happening? All the cell organelles, ribosomes, Golgi bodies, endoplasmic reticulum, mitochondria, as well as the microtubules, these are drawn to this area, to this equatorial region, this region. And what happens here later on? The Golgi bodies, they generate multiple fluid-filled vesicles that fuse with the growing cell plate, okay, and helps in the extension of the cell plate. Here you can see these are the vesicles, okay. These are the vesicles. They fuse with the growing cell plate. This is the growing cell plate. Vesicles fuse with them, leading to the extension or the extending of the cell plate. And finally, a complete cell plate is formed between the two cells. These are the two daughter cells that are formed. So now sometimes what happens, in certain areas, vesicles, they fail to fuse. And what happens? If they fail to fuse, okay, then this leads to the formation of plasmodesmata. And the cytoplasm of the neighboring daughter cells, they remain in contact with each other. Plasmodesmata, you know. So if this was the cell wall, here is the opening, okay, that connects the two cells. This opening is known as plasmodesmata, also known as the cytoplasmic channels. Through these channels, two cells are connected with each other. So what is happening here? Sometimes the vesicles, they fail to fuse, okay, and leads to the formation of plasmodesmata. So these are the stages of the mitosis, okay. Mitosis consists of interphase, and then we have the karyokinesis and the cytokinesis. Karyokinesis, which is the nuclear division, is divided into prophase, metaphase, anaphase, and telophase. And after karyokinesis, we have the cytokinesis stage, okay, this one, which leads to the formation of two daughter cells. So in the end, what is happening? There is formation of two daughter cells with identical genetic makeup. And this is also known as the equational division. Why? Because there was the division of the centromere, okay, leading to the separation of the sister chromatids. Now let's talk about the significance of mitosis. What is the importance of mitosis? So mitosis, that usually occurs in the somatic or the vegetative cells, helps in the growth and the cellular repair of the multicellular organism. Okay. Another point is that mitosis is the basis of the asexual reproduction. Okay, and helps to increase the number of the unicellular organism. Mitosis is also helpful in vegetative reproduction. Vegetative reproduction is also a type of asexual reproduction. Okay, so vegetative reproduction also takes place with the help of mitosis. And the most important point here to remember is that the chromosome number as well as the genetic constitution remain the same because of mitosis. Okay, chromosome number remains same. That's why we call it as equational division. Equational division. Okay, so this was all about the mitosis. We'll be studying about the meiosis in the next stages. Thank you very much.